And how about the informatics in Biogen?
Yeah, informatics is pretty good. They have a solid AI team also who works on it. A lot of artificial intelligence too. We
Mm,
have some collaborators, I think, from Harvard, MIT, and Carnegie Mellon too, who works with us on
okay.
that line. So it's a pretty good segment of it, but that comes way earlier in discovery phase. That
Mm,
I don't work with those interface
mm,
mechanisms mostly, but I mostly work on the ones that are further down the pipeline.
mm,
But yes, bioinformatics becomes like a fundamental backbone in
mm,
terms of drug discovery. There's no doubt in that. I don't think you can test 5,000 compounds directly in cell culture, right? So you got to start somewhere.
mm,
So
mm,
bioinformatics becomes a very, very, very uh,
mm,
fundamentally sound and also
mm,
very innovative way of doing fast research. So it's very helpful.
mm. So you're working basically in control, quality control or quality assurance?
No, 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 not both. I work on toxicology segment of drug discovery, so it's pretty much
Okay. Oh,
you'll see a segment of the time flow here. It's a little bit
yes,
different.
yes, yes.
We go in
Ah,
accordance to the FDA's
mhm,
application formats.
mm,
I worked with
mm,
OECD too, which is mostly European, and also the MFWH, which is part of the Japanese segment, and
mm,
some of the Asian countries too. I'll probably talk a little bit of everything, just like a
mm,
highlight points on what I've done, because
mm,
I've previously study director for DART and juvenile studies in Charles River. That's
mm,
as a senior study director for about 3 years before I moved to Biogen. So there was
mm,
a lot of that was hardcore preclinical. Like
mm,
that's very timeline inducive. So you'll have often FDA folks come into
mm,
your office to check
mm,
everything, including your computers and printers.
mm.
So it's a
Okay.
pretty yeah. So
Mm,
it's a little bit of everything. But I think you'll today when I go through the flow chart right from phase one
mm,
all the way through your final clinical stage, I think it'll be a
mm,
better introduction for everybody. Also, I think that's probably why I suggested this topic
mm.
because I'm not an expert in bioinformatics. Neither was I one in the past either. So
Mm.
I don't think I would have the
No,
forte to talk about
this
that
is an
per se.
amalgamating subject. We can link with all the
Correct.
subjects. Yeah, drug discovery is a
Correct. Correct.
common one.
Correct.
Yes.
Correct. 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 Yeah, it's a little bit of everything. So I'm really excited about it.
2 months before, I also find an advertisement from JRF Global for toxicologist. So they are
Okay.
providing here, it's in Gujarat, they are providing nearly
Okay.
minimum 18 lakhs per annum.
Oh wow.
Yeah, it's a great opportunity. They start from
Nice.
18 lakhs per annum. So it was just a
Nice.
toxicologist position. I don't know at that time.
Mm-hmm.
So they're
Mm-hmm.
asking like, do you know any toxicologist agency in USA? They ask few questions related to toxicology kind of. So
Yeah.
who
Yeah.
has
Yeah.
to give approval? So if
Yeah.
I
Yeah.
might have
Yeah.
met you earlier, I might have cleared all that
Yeah.
questions. Yeah.
Yeah, that's a
Uh,
whole new line of topic
mm,
about drug application. If you guys want me to
mm,
talk about that at later
mm,
point of the
mm,
year, I can do that too.
mm,
I file
mm.
a lot of those applications. I
Oh,
used
ho,
to in the
ho.
past. I still
Mm,
do at some points. Now we
mm,
have folks who do it for us. I
mm,
don't per se.
mm,
Mine is a
mm,
more subject matter expert
mm,
consulting type of work. I
mm,
don't involved in the wet lab research or
mm.
any of the animal research or any of that
I
segment
think
of
your
work per se.
younger brother is also working as a scientist in USA, isn't it? So close to you.
Yeah. He's not a scientist. He works actually for Walmart. He's a
Okay,
senior product manager for Walmart. He used to work for Samsung at Mountain View in
okay.
California. Now he moved to Walmart. I think maybe a few
Oh,
months
oh,
ago.
oh,
So he's a non-engine. He's an engineering background, non-scientist background.
oh,
But
oh, oh, oh.
he did his masters and everything from Purdue
Okay,
in
okay, okay.
Indiana, at West
Yeah.
Lafayette, Indiana. So that's how he's in. He's more the
Mm,
analyst and e-commerce expert for Walmart at this
mm,
point.
mm.
Yep. So, Kumar, quick question. This might be out of my ignorance. I didn't look at the website recently. So, the head of the department, I
Is
think
Dr.
somebody
V.
talked.
Smita. Dr. V. Smita.
Oh,
Yes.
wonderful.
Hi.
Hello. Sorry, I did not recognize the voice.
Not at
My
all. No
bad.
problem. No
No, no, no,
problem.
no.
And
Now,
really, I was really happy to see you seeing and
thank you.
talking. And I was just listening to the way you were talking and you were addressing to Praveen.
Thank you, thank you. No, no, all that I remember about, I want to say Sumita ma'am because that's kind of how I know it. I know Praveen sir too, from the past, of course. I only remember the downstream processing lab. There's nothing beyond that kind of hits my brain. But I really enjoyed all those times. Thank you all. I really appreciate it.
Thank you, Aditya.
Is
And
Dr.
we
Shiva
are
on the line too, or?
really no,
Yeah, go ahead, please.
sir's not joined the meeting. He's there at the trust office as a secretary of Sri Venkateshwar College of Engineering and Health.
Fantastic.
So,
Yeah, I was in correspondence with him. I was working in Charles River. I corresponded with him for something a couple times, and then he said that we will meet when I come to Chennai, and it's been 2 years since we talked about that. I don't think the pandemic let me or my family members to come to Chennai at any point recently. But next time when I can, I'll try to stop by with him too. We wanted to have a quick chat on something real quick. But I really appreciate Dr. Sumita, Dr. Praveen, and everybody else and the rest of the team who organized, and the folks who are online. This, I think, will be a good opportunity for me to interact with you guys. And also, anytime during the presentation, anybody, I'm just giving you a heads up, do not hesitate. Just interrupt as you can, because the way I see things is that no question is a stupid question. So feel free to enjoy and interact as much as you can. I think we still have a couple minutes, so I'm just going to hold online until then.
Aditya, Prabha ma'am is also online. Prabha ma'am is also here.
Fantastic. How are you doing, ma'am?
She's assistant HOD, Dr. Aditya.
Fantastic. Man, I'm really excited now. I think I can talk to some of the older folks too. I think it's a good time to reconnect with everybody.
We'll be happy to have you in SEC once you're here in India.
Yes. Sometime this year, hopefully mid-year, if things go well and if God wills me to walk back to India sometime, I'll definitely stop by, and we can definitely discuss a few sessions at that point.
Yeah, plenty of opportunities are there in the area of topic what you're going to take. In India, so many opportunities emerged in this field.
Yeah, I saw some recent government ads too, in terms of biotechnology investment, and I think several years ago they put up one, and I think one of my family member took a big advantage of it, and now they are not in the biotech field but something else in the plastic field. Now they're big in the business. I think that's great. Finally some shed of light. That's really exciting.
HOD, madam?
Yes, Praveen,
can
sir,
we
can start. No, a few others let them join.
Harini?
It's already 9 o'clock. We'll start.
Yes, ma'am. I also started recording. Harini, are you there?
Sir.
Can you start,
Sir,
Harini?
Madhu will be doing the introductions.
Madhu, are you there?
Yes, sir.
Yes, you can start, Madhu, now. Good morning.
Okay. Good morning to one and all present here. I hope you're all safe and doing well. I'm Madhumita from second year biotech, and I am extend my warm welcome to our head of department, Dr. V. Sumita ma'am, our assistant head of the department, Dr. S. Pandiprabha ma'am, our esteemed guest, Dr. Adithiya Charli, our staffs and students on the behalf of the Department of Biotechnology and Bioacademy to today's biotalk on the world of drug discovery. Brief overview from the drug discovery to deliverance to the patients. Today we have with us Dr. Adithiya Charli, who is senior scientist, Biogen Cambridge MA. With 8+ years of experience in toxicology, Dr. Adithiya Charli began his journey with BTech Biotechnology at Anna University and Doctor of Philosophy at the State University. Dr. Charli worked as a senior scientific associate, Charlie's River Laboratories, and experienced in the field of developmental and reproductive toxicology. He currently works as the senior scientist Biogen Cambridge MA. He also serves as a senior counselor for the Association of Scientists of the Indian Origin at the Society of Toxicology. Dr. Charli is also has won for several prestigious scholarships and awards like Postdoctoral Research Award 2018 is instituted by the College of Veterinary Medicine at State University. Dr. Charli has also published several papers, is a member of organizations like Society of Toxicology and American College of Toxicology, and and we are happy and proud to have you here today, sir. We hope to learn a lot from you. Over to you, sir.
Thank you, Madhumita. I really appreciate the introduction, and thank you. And it's good to, at least virtually meet, all my alumni friends and also some of my faculty members. Really appreciate the opportunity. Dr. Praveen, Sumita ma'am, Pandipra ma'am, it's really an honor to be back virtually, at least. I think that's the only way to meet nowadays for you guys. So the topic I'm going to be discussing with you is some of the lines of experience that I've had over the past several years, working as a study director, working part of Biogen, which is one of the biggest developers of neurological drug models in the United States and across the world. And also some of my other research experiences that have, legibly allowed me to transit over the years in the field of toxicology. So I'm just going to give you so everybody can see my slides, right?
Yes, Aditya, we're able.
Yes, sir. Yes, sir.
Perfect. So I'm just going to do a quick outline about my academic background and my research background that would kind of direct or tunnel me towards the other topics of discussion of drug discovery and whatnot. So I'm just going to go over that. And then also, of course, the main topic about drug discovery from, all the way through the deliverance to patients, which is ultimately what's happening to every research or drug model that we do. It has a commercial viability, and also from a personal front, it helps the life of a patient, which is definitely the ultimate, success in the field of drug discovery or any type of drug model per se. So I did my bachelor's at Venkateshwara, worked with some of the great scientists there and faculty members. I think Dr. Shiva has been somebody who's been very phenomenally instrumental. So definitely all his sessions and every other session was definitely very edifying for me personally too. And I moved to the United States to do my PhD in toxicology. And I got my PhD in toxicology, minored in neuroscience, and also in molecular and cell developmental biology, and also in molecular genetics. Then most of my research was all on neurotox, mostly Parkinson's disease and Alzheimer's. That was my backbone and background. And as a postdoc, I worked on mostly translational and neurodegenerative models and biomarker drug discovery based models. So I was basically at Iowa State for over 6 years, doing my PhD and my postdoc inclusive. And I mostly did my work in the veterinary medicine college. So I'm so used to working with small, medium, and also extremely large size animals as big as horses. I've done a lot of horse work, cow, pigs, I think mice, rats, rabbits, dogs, and also primates in some cases. So that's all been very phenomenally instrumental in shaping me in where I am at this point. And thanks to some of those opportunities, got some nice chance to publish a bunch of papers in some of the high impact journals. I have some in Nature, probably Science Letters, Translation Medicine, Journal of Clinical Investigation, and also some coming up in Nature Neuroscience, which is in the pipeline for some of my later PhD work. It takes a long time, so I can't help that. And I also had some chance to publish a few book chapters too, and also work with some of the scientists on that front. And I also serve as a manuscript reviewer and also an expert advisor for the Toxi, which is a toxicological sciences journal, and also for the ACS Chemical Neuroscience Journal. I've been doing this thing for over 3 years now. So that's been a great experience. And prior to me being currently at Biogen, I was actually at Charles River, which is one of the biggest contract research organizations on the planet. And I think they preclinical test or safety test for about 80% of the drugs that come to market from the United States alone. So that's the level to which that CRO has functioned. And I think I worked on most of the DART as in it means development and reproductive toxicology, and also juvenile toxicology. So all your vaccine testing, there's some of them that I've personally done for some of the companies that that recently does the vaccine work for the United States. I've done a lot of phototox and gene therapy work also, and everything are all aligned with GLP, non-GLP guidelines and in accordance to the FDA, which is the Food and Drug Administration segment, and also the OECD, which is part of the European drug segment. And also I served as some of the panel members and site lead for global quality plan for Charles River. And then I moved to Biogen recently, maybe a few months ago, as a senior scientist, drug discovery toxicologist for being a subject matter expert mostly. That's probably what they wanted me to do mostly for DART juvenile neurotox models, and also gene therapy and some of the other therapeutic targets like ASO and small molecules as well. So that's my current work at Biogen, but I'm just not going to bore you guys too much with that. I'm just going to give you some brief outline on where I started, what I've seen through the years, and what's led me to be where. So my background itself, from my undergrad has always been in biotechnology. I think I had an elective in biotechnology in my high school. Then I did my undergrad from Venkateshwara, did my doctoral research, my postdoctoral research. Everything was around the segment of neuroscience and a lot of neuroinflammatory toxicology work. And then later I settled down in the preclinical and also work on a lot of clinical safety regulatory studies for Biogen currently. So yeah, my graduate school was in Iowa State. It's a great place, one of the best veterinary colleges, one of the best schools for biomedical sciences. And during that time, I think I had a lot of collaborators with whom I worked from Caltech and UPenn. It was one of the bigger segments. Harvard also. And I also had a collaborator from Oxford also at that point. So it was a pretty good experience working in Iowa State. And I was actually very happy to have a very fantastic mentor who's currently moved to another university now. But, he was a fundamentally very supportive person during my PhD. And a brief outline on my doctoral work and postdoctoral work. I'm not going to give you all the mechanistic details of it because I think it might be a little bit of an overkill. But if you have any other specific questions, always don't hesitate to interrupt in the session right now, and I'll be more than happy to stop by and give you my explanations on those. So I've done several models, more work. So when you say any type of drug discovery or any type of research, there are three types of models that goes into place. Either it is your in vitro toxicology work or in vitro models, which is basically a cell culture, which is your immortalized cell lines that you work with. In some cases, people do work with primary cell lines too, especially if I'm going to work on a lot of neuro cultures or neuronal cultures. Since I do most of my work on neuronal research or neurodegenerative diseases, that's pretty much what my length of work lies. And also the next facet comes about your in vivo work. What do you do with your animals? If it works well in cells, can you actually replicate the same concurrently and also in some cases repetitively in animals? And then the last facet becomes your clinical sample investigation. So I've done several cell culture models. I don't even know the number of cell lines I've worked with, but I've done several, several hundreds of them maybe. I've also created a lot of cell lines for myself, which are using the CRISPR-Cas technology. I think recently, maybe if you look at the news, everybody uses something called CRISPR-Cas to knock out a specific gene expression or also to overexpress a cell line. I worked on a lot of those technologies over the years. I think even now, a lot of my younger lab folks, graduate students who I've trained personally, have also had a lot of chances to work on these cell lines. So a lot of these signaling cell lines I've worked on over the years, and then also had a lot of chance to work on a lot of animal testing. Thanks to my experience with the IACUC, which is the operational unit that functions to withhold and format animal welfare. I've also worked with a lot of animal studies, done all types of dosing regimes. In toxicology, the route of administration or the route through which you inject into an animal is very critical. I've done maybe oral gavages, subcutaneous injections, IV type of injections, lateral tail vein injections. I've also done a lot of stereotactic surgeries in which you can open the skull, the brain of a live animal by keeping them in anesthesia, and then later trying to inject some viruses into it for checking certain expression levels, and then again completing the surgery with closing those open surgery marks and then checking and monitoring animals for a few months. So I've worked on those for rabbits and rodents also. So those are some great experiences with animal models, and also done a few ex vivo pup models, which is called the organotypic slice culture model, which is definitely very, very famous. I think it's famous across the world, not just the United States. That gives you a lot of leverage to minimize your animal work, because as question comes about why do you waste animals, or why do you use a lot of animals, and you always have the backbone to say that you've done a lot of these subcultures of organotypic models to refrain from using excessive number of animals than what you might actually need to use for a study. But this, at the end of the day, you do have to do animal work to get past all these mechanistic toxicology studies. Then I also worked on validation out of human samples. I have some collaborators back in South Africa, some out in the Western European segment too, who sends in samples from clinical or from postmortem brain samples, in which we test for markers of Parkinson's. I think I've done Alzheimer's. I've done folks with traumatic brain injury too. Several of those fragments, and I also worked on some of the models to discover biomarkers, which is currently under a intellectual property patent work with my previous boss during graduate school. So that's going on well too. So those are some of my overarching work. I've done a little bit of everything to understand where or what I want to do the next step of my research. Thanks to some of all my research experiences and everything, as I've gotten some opportunities to publish manuscripts, work on scientific presentations, received some scholarships and awards at the national level and also at the graduate school level, published several of the high impact journals, mentored a lot of students, which is definitely I liked doing that a lot personally, because it gives you a lot of leverage to understand everybody's point of view in science. I've trained undergraduate students, veterinary students, and also doctoral students during my times as PhDs and as a postdoc. And then I've been an invited speaker for various conferences and scientific platforms over the years, and also worked on a lot of bigger grants like the National Institute of Health, the Department of Defense, the Michael J. Fox Foundation, and various other segments in the United States. A lot of these are all the bigger funding agencies with whom I worked over the years. A quick outlay on a few studies that I've worked on. I don't want to again get too much into the detail, but I want to give you a high level subject matter information about to the level to which you can actually perform research as an individual at the doctoral level or at the postdoctoral level. Because when we say drug discovery, you actually want to find maybe a receptor-based target or any set of a target for a disease at the molecular level to see if that expression of the disease manifests itself or propagates, or if there's any sort of disease prognosis towards the end of, whenever you want to complete the process of exposure. Exposure is a common word in toxicology. So that's kind of what you see as an endpoint. For example, in this study, this is actually, I think it was an NIH grant worth maybe $2.5–3 million or something when I was in graduate school with that I worked on personally. And we were very glad to work on this because it was kind of a very dry state at one point when we really had to get that money to do some research. So this work was about how environmental pesticides, when you say neurodegenerative disease, one of the biggest villains that causes those type of diseases like Alzheimer's, Parkinson's, Huntington's, ALS, MS, muscular dystrophy, various other traumatic brain injuries, something to do with spinal cord injuries, and you have a lot of these nerve damages. All of these at some point is caused due to exposure to environmental hazard agents, pesticides, some cases well water, some cases metals, especially divalent heavy metals, causes a lot of neurodegenerative diseases. So the bottom line is to see how certain environmental pesticides, which comes under the class of mitochondrial inhibiting compounds, in other words, that's pretty much cutting out your respiration for your cells. How does that trigger an epigenetic mechanism in which you can see modifications on the histones, which are actually within the outer surfaces or the crevices of the DNA, or that they form as this, actually like a backbone for your DNA structure per se. How does that affect, and how does that in turn cause any type of gene alterations? I did some high-level imaging on this one, a bunch of animal studies, and all of these gene targeting studies that you can see in which you can check gene transcription, translation level modifications. So this was one of the mechanisms that we thought actually triggered, because if you think about a cell structure, you have a nucleus, you have mitochondria and other organelles lying outside the nucleus. But if you actually hit the mitochondria from outside, it actually triggers subnuclear changes, which is kind of very surprising because many people at that level did not know that it existed. I think we were one of the first few groups to break that taboo on how mitochondria actually affects the nucleus, and you just can't say it's a carrier or a receptor based aggressive toxicology output. So we demonstrated that, and that was very much of a encouraged output at that point. Another bigger research that we did was partially of what I told, how mitochondria affects the nucleus, but we also showed that in another mechanism that it can actually trigger a variety of kinase activity, because if you think about a big cell, kinase is one of those proteins or enzymes that run around inside, and they have a very aggressive mechanism of action. They basically, once they get activated, they basically phosphorylate everything and anything that they basically see. In most cases, they're very specific. So I found a mechanism in which it actually denatures or damages the nuclear membrane also. So it's more like an elastic tug of war moment. When you actually mitochondria damages, it actually rips your nuclear membrane apart and then stops to collapse all the internal segments within the nucleus itself. So it's a very interesting segment. I think I did a few live imaging to show how quick and sequentially this process happens, and I think that was also a very fruitful output. These are something that I think I had in my doctoral thesis a while ago. This is, I think, currently in publication or in review with Nature Neuroscience, and I think the previous one that I showed, I think we are targeting to put that through clinical investigations, journal of clinical investigation. But apart from just the mitochondrial output, I worked on other neuroinflammatory targets. I don't want to get into the details of it, but I'm just going to brush over a few things, something related to potassium channels, destruction in neuroinflammation, some markers related to NLRP3. I don't know if you guys get a chance to read any journals, all through the past 3-4 years, you will always hear the word inflammasome, which is becoming a big target in terms of drug discovery, in terms of academic research. It just excites everybody. It's one of those targets that affects your astrocyte or your microglial population in the brain, which is basically functioning as a resident macrophage, which is actually a type of an immune cell. So they do have emits C receptors and subclasses of those as well. Another one is about how this protein called alpha-synuclein, which is one of the markers of Parkinson's disease, wherein you can see this protein. So originally the way it was actually discovered is they took the head of a post-mortem person who died of Parkinson's disease. They dissected the brain and figured that there was a small patch or plaque-like substance accumulated on different regions of the midbrain that spread all the way from the olfactory all the way through the pons and medulla. And people identified that to be a bunch of aggregated misfolded proteins, and those are proteins are pretty much what's called as alpha-synuclein, and the important component is actually called alpha-synuclein. That cluster is actually called cellular plaque, something like what you see in Alzheimer's too. So we found some mechanisms in which how this plaque gets transported and causes all sort of toxicity models. So I was mostly involved in finding mechanisms that causes cell damage instead of just the cell rescue portions of it, because if you can find the cause of a damage, is always easy to find the rescue for the whatever damage you're looking for. And these are some of the funding agencies that I probably previously talked about that I worked with as well. And you can see a picture at the back. That's actually a moving version of an entire mouse neuron that I captured. I think it was using a 3D confocal microscopy or something like that. You can actually see all the neuronal connections that established through these neurons with the other neurons in the brain as well. So this is an entire bunch of, I think, 5 or 6 neurons, maybe close to 5-6 microns in scale length or something like that. So this is pretty exciting that worked on. So overall, it was more about neurotoxicology mechanisms and not just a drug discovery mechanism. But I understood the value about how do you dissect out each mechanism to find out what you're really looking for. So that becomes the fundamental basics of drug discovery. And then once I did my postdoc, my PhD and my postdoc, then I moved on to Charles River to work as a study director for several years. I worked with some of the biggest pharma companies in the business. That includes Pfizer, Merck, AstraZeneca, Biogen too. Before I joined here, other folks were AbbVie, Genentech, and I worked on a lot of different studies for most of them. including some of the recent vaccine work and COVID medicine testing too, so for Pfizer and Merck. So just moving on to that, after Charles River, that was in Pennsylvania. So I've lived in Iowa for most of the time, moved to Pennsylvania, from Pennsylvania now, and I'm in the Boston area at Cambridge. And this is actually an overall view of Cambridge. It's a beautiful, beautiful city. It's part of Boston. So Biogen is situated right in the dead center. On the left-hand side, it's Harvard. Right-hand side is MIT. Center, about 10 or 15 buildings is all Biogen buildings. We have other companies who are situated around this area too. So I do have a lot of collaborative work with Harvard and a few of the other medical universities like Tufts and other folks too ongoing. But I'm just going to get you through a little bit of how the real drug discovery process starts and how the process actually ends. But before we get to that, there are a couple of terminologies that you might want to be familiar with, which I'm going to be repetitively using just for that. It's just a small bit on this. This is a saying from one of the researchers maybe 5-6 centuries ago, who talked about all substances are poisons. There are none which is not a poison, which is actually true. The true dose differentiates a poison and a remedy. So in a shorter version, what you say is a dose decides if a poison is actually a poison or not, pretty much. So there's always this dose effect causing toxicity or cell death, and what is the longevity through which you've exposed to that dose, or what is the frequency through which you've exposed to that dose, is again that what decides toxic effect of any compound per se. Here is actually a funny analogical table. At least I find it to be funny. So botulinum toxin is definitely a plant-based toxin, or in other words, it's a natural-based toxin. Sorry. So if you see the dose levels to which it's actually toxic is extremely low, which means it's really potent, right? But when you see lead, and which is again a divalent metal, which caused a lot of problems over the years in the United States and also many other part of the world too. That's called lead toxicity, which has resulted in a lot of juvenile folks getting affected, especially the pediatric population. So that's got some levels of lethal toxicity. There's another model that everybody's quite familiar with. It is apart from lead, you would have heard about the mercury toxicity, Minamata disease, right? So that's again one of those. This is on similar line. But you might wonder, why is vitamin B6 in this list? Because vitamin B6, for all that was taught to us right from kindergarten all the way through high school and college, is that vitamin B6 is very important, it's very good. But it's only good at the levels it's supposed to be taken per day. But if you go overboard, it's going to be really toxic. Toxic per se, it could cause a lot of ataxia, which is ataxic. One thing is the neuropathic changes, a lot of gait mechanisms being hindered, all those things you can see sometimes in vitamin B6, but it's not something that you see common, because at what dose you're going to take vitamin B6. Again, the most funniest thing is water. Everybody want you to take few liters, 2 to 3 liters of water each day, which is approximately a gallon, which is 3 and 3.5 liters of water each day. But if you're going to do overdose of water, you might actually have a lot of side effects. I don't know if people actually believe it, but I think that might be true, because there is a recent work on green tea, right? Every one of us have green tea, right? Everybody tells, oh, green tea is good for your body health, your nourishment, antioxidant. It helps to, keep your immunity high and all this stuff. But there's a specific compound in green tea called EGCG. I think I worked on that one too at one point with one of my Chinese collaborators from Shanghai. She's actually a food toxicologist and also a neuropathic scientist. So she would do a lot of work on green tea too. So only certain concentrations of green tea or the chemical in green tea is needed each day. For example, up to 5 millimolar each day is apparently needed for antioxidant effect. But if you want to take 5 millimolar of EGCG alone, that's 5 millimolar. But if you want to take it along with the components in green tea, you might land up drinking 10 to 15 liters of green tea, which is going to be detrimental to your liver and your tummy. So apparently it's actually a toxin if you toxin if you actually take it more than the required quantity. So the bottom line is dose makes toxic effects. Sorry. So this is again, a big compound. The reason why I'm showing you this information is so that you understand that there is a specific rule by which the FDA functions, and how do you take your drug from discovery stage all the way to your patients in order to give that cure or treatment or prevention of disease. But along this way, the way they have designed this thing is such that they have seen a lot of catastrophic effects because of drugs that's happened in the past. For example, this wonder drug, as they call it, is actually they call it the elixir. Elixir is not a chemical name, but it's an elixir because it apparently cures everything. With some type of sulfanilamide, which is apparently was actually used for treatment of any type of infection, mostly a streptococci type of infection. But apparently they figured that this was some sort of a wonder drug. But what it actually led to was death of about 71 adults and 34 children, which is a lot, which is actually catastrophic hell when compared to the number of deaths it's caused. So then the government and agencies and everybody started to form, and then one thing left to the other to decide on how do you structure all these. For example, another biggest example was the thalidomide. Thalidomide was supposed to cause or prevent nausea for women who are pregnant. That's why the drug actually came to market. This was maybe a few years ago, not a few years ago, several decades ago. But once the drug came, it did what it did. It's supposed to block nausea, feeling for pregnant women. It did. But the problem was that the infants who were born were really, really had a lot of congenital issues, which was really bad. I don't want to bring the pictures up. It's going to be really horrifying to look at those pictures. But since I came from a developmental tox background, I know those work. And that again is where a drug has gone wrong because people didn't do enough safety testing on it, right? So then the Congress, the United States, started to compile and bring in slowly the drug acts all together, the food, and that's when you started having the birth of the FDA, USDA, various other divisions to prove your food quality. And then one thing led to the other that you started to increase your level of toxicology testing. So historically, there was preclinical testing was done. There was a lot of toxicity continued to cause primary attrition. In other words, of what we just talked about. So they had a lot of projects open. There were a lot of issues happening, so they had to close one over the other to the other to the other to figure out what was the best method to actually execute tox testing. Because those were the times, think about it, 50, 60 years ago when nobody had the exact mechanism of action. There was not enough of testing. There's not enough of safety guidelines, which we're supposed to be following. Because I worked on the safety field for a while now, 8-9 years. So I kind of know that there's a lot of safety guidelines. So for example, say you want to get one drug that is responsible for treating pancreatic cancer, right? So it's an oncogenic endpoint. It has to pass through a variety of testing, including testing in pregnant populations and at least in terms of in vivo toxicology testing. You got to pass through pregnant population, young population, like juvenile tox population, and all of those spectrums before the drug can actually come to market. And this was also done in conjuncture with a lot of in vitro model testing, clinical sample testing, and so on and so forth. In other words, this is a multifactorial process that does not have a linear timeline, but it's more multi-transcendence or transitional timeline per se. So the bottom line from this is that the information that need to be discovered during the discovery phase of a drug started to gain more importance and traction because people started to find issues with it when it actually going to the clinical sample or clinical patients, because that's not what you want to see, right, at the end point. So we started to see a lot of effect and stringency in the rule even earlier in the spectrum. So now we come to the real big space, right? So when we say preclinical testing, which is preclinical time, what are the things that we look into? It's primarily made up of two segments. One is the discovery toxicology group, and the other one is the preclinical and clinical toxicology. So I'm just going to go over the timeline with you guys. It will be more fun to look at. Oops, my bad. All right. So if you see discovery toxicology, it's got three hallmarks. One is the target discovery, other one is lead identification, the next is the lead optimization. So when you say target discovery, pretty much what you're looking at is you want to have a, say for example, you want to find a drug for Parkinson's disease. So you want to know what is the target that you want to do to minimize the effect that causes prognosis of Parkinson's disease. Say for example, you want to hit a target that's a receptor and you want to make the receptor's activity mute. So that's one of the targets. I'm just giving you an example. So you find a target and make sure that that target fits in with your drug model of its drug models per se. For example, you're looking at a small molecule, say to bind with the receptor and function as a receptor agonist or an antagonist, depending on whatever function you want to do. In that case, you want to have a solid in silico model first established. I think this is what Dr. Praveen Kumar talked with me before we started this call about how do you use bioinformatics. So there's a lot of in silico analysis to especially clear and clarify drug targeting and drug at least your target discovery type of models. Some of the studies that you'll see is your docking study. Some models you'll see is a lot of these. Nowadays, we use a lot of artificial intelligence models to per se model whichever compound you want to look at. And then from there, once you know what your target is, you want to start finding what your lead identification is. So in terms of what are your molecules going to do? What are the studies that you're going to work with those molecules? What are the in vitro toxicology screening that you want to do? For example, you're looking, as you said, working on Parkinson's disease, then you want to get dopaminergic cell line that will actually function as your in vitro model, because you want to work on a cell line in which you put your compound of interest to see if there is any kind of toxicology effect. In other words, you're basically measuring cell death in order to see if there is any sort of effect of your compound to your target, whatever your target might be, neurons, neuronal cells, any other cells. But in this case, since I'm talking about Parkinson's, neurons probably would be the best segment to work with. And again, to see what are the safety markers that you can actually establish. Some of the times we work on a lot of these cytotoxicity assays, various other non-GLP type of designs to see if your compound interacts with your cell of interest, what are the other effects you see. So this is one time when you actually try to look at a lot of off-target effects for your compounds as well. So that comprises for your lead identification and lead optimization. And then the next is your transitional point. Now, by the time, this is already going to take you several years to get here. It's easy to say the way I'm saying it, but this is going to take you at least, depending on your compound and everything, at least, 4 or 5 years to get here. All the way from what your target is, all the way to screen a compound out. It's going to be that much level of changes that you're going to see. Another interesting thing here actually is that when you actually have a drug target and you're designing a compound, you're not going to design one compound, because the company is not going to invest several $1 million, tens and 50s and hundreds of millions of dollars just for one compound. Because what if this one compound goes all the way through clinic, say it's a very lucky, a magic drug, and goes to clinic, but then fails in clinic. So which means we lose $50-60 million just betting on one horse for the race. So what we do is we actually try to create hundreds of targets or hundreds of compounds, and you start to screen with compound 1, 2, 3, 4, so on and so forth in all your in vitro models, and then pull out a lead model. So lead means a compound that's come down the funnel, checked all the check boxes back and forth for an n number of times for any number of investors to see. And then you have a candidate selection that will go into what is called as R2D, which is research to development transition. This central union point is the biggest trump card. And people like me actually work in this segment mostly, who know the preclinical world, clinical world, and also the discovery space. We are in charge of working on all these compound leads and lead tox studies. So next comes the fun and the most exciting work that we do with our colleagues and friends at FDA, in which you start to do a lot of these safety studies on animals. Now you've passed your cell stage, you have a compound, you do a lot of safety studies on your animals. So when you do safety studies on your animals, depending on what disease, you got to work on which animal model which is probably one of the most chaotic things that you can ever work on, because when you do mice, rats, it's fine. Rabbits are fine. And when you start to move towards the GLP or the USDA regulatory species like NHP, which is monkeys, mostly macaws, depending on what species we want to work on, dogs, some cases pigs, it depends on which model again. So those becomes the more highly expensive studies. For example, a single monkey study can actually cost us close to $250,000 just to buy the animals alone. Just the cost of a bunch of animals is going to cost you $250,000 to $500,000. And apart from that, you're going to start working on the compound, the drug, the study, all the labor, everything. It's going to cost you several $1 million to do just one single study. So that's the level to which you precisely program what studies you want to do and what population of animals you want to do, what sex of animals you want to do, and what age of animals you want to do. Because age, sex, population, species, strain makes the biggest decisions ever. So I work with a lot of those type of study designs as well for the most part. But since I've had a lot of preclinical experience in this in the past, working with those designs is actually more fun to me than trouble. And then once you pass all these in vitro and then in vivo stages, you push a compound all the way through clinical phases. When you start to go through phase 1, 2, and 3, it's a clinical phase. I will get to the details of it in the next few slides to come. Excuse me. And I think you'll see a little bit of more interference on what you want to do with those type of studies. So for example, think about it this way. Phase 1, you're going to use less number of human beings or less number of human volunteers. Phase 2, you're going to employ more number of human volunteers. And then phase 3, you're going to employ a bigger population, a few thousand human volunteers for testing. This is the way your vaccines are done, your small molecules, your tablets, gene therapy, whatever drug testing that you want to do. But along in each phase, apart from just working on those population, you also need to satisfy a lot of subcategory studies. For example, in the phase 2 and phase 3, when clinical studies are going on, you also need to simultaneously do a lot of in vivo studies, which are GLP quality, and try to satisfy points such as reproductive toxicology, carcinogenicity. Because once you go to the final human population, which is your registration, you need to have all this data before the government lets you to actually register a drug to produce mass levels of it at the GMP standards. So there should be a lot of studies that needs to be done throughout clinical. So clinical and preclinical will happen simultaneously before you can reach the endpoint of clinical. So this is what I was talking to you about. I think I briefly highlighted everything, but I'm just going to go over the quick details. So this is the first phase of what we saw, discovery talks, right? So you have a target discovery, you have a hit ID, you screen for your lead compound, which is one of your compounds, and you optimize your lead, make sure that you do all the sub non-GLP studies before you can take it to R2D transition. So mostly in your you do target assessment, you do a lot of in vitro testing like cytotoxicity, gene toxicity, in vitro hepatotoxicity, phototox testing. You do a lot of ion channel testing too. And then you do some early animal studies to see if a specific compound, but probably 2 or 3 of them individually, do they cause any sort of strain on your animals? And if not, then they pass. And then you go on to the next set of studies. And then in terms of animal studies, I think I told you everything, right? One of the other things that we need to talk about is the safety margin. What concentration of a drug is safe? And what is that margin categorized for? So that's the question. So in other words, you're going to decipher something called as the NOEL. It's called the No Observable Adverse Effect Levels, which will tell you to what concentration, when you put it into an animal, you see no change. When you say change, what you're looking at is body weight. You're looking at clinical observations. You're looking at the food consumption of animals. You will look at the behavioral aspects of animals. And you will also see a terminal necropsy with look into microscopic and macroscopic pathological reviews. So that's what will determine a toxicity level for an oil. So that's pretty much what these graphs have in mind. And then ED here refers to the effective dose, a dose to which you can see maximum exposure, but minimum toxicology effect. So that is the sweet dose that we call as a clinical, that you can translate to a clinical dose eventually when the time comes. Because you want to take a tablet, say for example, you have a headache, you want to take an ibuprofen, you take ibuprofen, you can go up to 600 mg a shot. Till 600 mg, you know it's going to be foolproof, but the window will keep it up to 1000 mg because you don't want to overconsume the drug even by chance. But if you do anything beyond 2000 mg or 3000 mg, which is 3 grams, that's a lot of drug abuse. That's a drug overdose. So you decide what maximum concentration that the body can assimilate and dissipate causes the maximum effect for the disease, or if in this case fever, cough, what are we looking for, treatments that can effect for. But you don't want to see anything beyond that. You don't want to see side effects. So you don't want to take a paracetamol and see side effects, right? So you want to take a paracetamol, get off your cold, fever, or your headache or whatever, then you want to move on, and you don't want a drug to reverse back, or in other words revert and then have side effects on you. So that's what you look for in oil, which is very important at the tox levels. And I think this is also something that I briefly talked to you about, species requirements. You've got to look at what is your compound. Is it a small molecule? If it's a small molecule, which is basically your receptor-based targets or something, what species you want to test, how long do you want to test, what type of testing you want to do. And if it's non-rodents, are you going to do NHP, dog, rabbit, or if it's going to be rodents, are you going to do mouse, rat, hamster? What are you looking for? And what is your target? You want to look for target expression, homology, PK, PD modeling, physiology and pathology and morphological differences. And also the biggest question, what is the route you want to administer? Most cases, animal study routes are decided based on what clinical routes you want to take. So that's what decides your animal route studies for the most part. And then just a couple more slides before we come to an end. Another thing is routine endpoints. As I told you, we look at clinical observation, food and body weight in animals. We look at toxicokinetics to see what exposure levels you can see at what time, where you actually measure the Tmax, the time duration to which you see maximum exposure in an animal. You see hematology outputs, RBC. You see an entire clinical pathology, clinical chemistry outputs, RBC, WBC, erythrocytes, leukocytes. You look at your eosinophil levels. You look at your protein levels, liver enzymes, so on and so forth. And you also look at a lot of histopathology, which is very, very important, because you might look at an animal, everything looks good. You look body weight, no effects, everything looks good to you. But if you look at it, your liver might have some sort of cirrhosis or some sort of lesions. That's something that you don't want to see. And those kind of testing is looked at. It says 20 organs here, but we hit up to 45, 50 organs. We check almost everything that's left in the animal body to evaluate and reevaluate that difference. And I think these are some details that I just went out of extra to talk to you guys about. But there is a lot of groups. This is an interdisciplinary business. We have a tremendous subordinate team. So I work as a tox lead for the compound. So I have my program lead, I have my chemistry expert, I have my pathology expert, I have my clinical doctors, I have an entire team of managers who work for data outputs and everything, and they bring it to me for review. There's a whole new bunch of phenomenal technicians who work on the process. You have a lot of folks from the iCook background, DMPK background, pharmaceutical industry, people who are from the formulations and manufacturing background. So it's this big group of people who work together all the time. But this group is actually led by 3 or 4 scientists who forms the upper ladder of the segment, which is mostly lead toxicologist, in this case me, a pathologist, clinical reviewer, and also some of the other safety concern officers if we need. They are the people who actually connect the rest with the team, either who's your compound leader or be it your program leader or whoever it might be, your RDPL or your research development program lead. So this is what I talked to you about. You start with 10,000 compounds, some cases even more. I'm not even exaggerating. You come down to only one compound. That one wonder drug is probably that's going to go to the market. That's what's going to help the life of a patient. I've personally seen the life story of a lot of kids and adults who have benefited with some of the drugs that I've worked at some point or the other. Recently too, right? Merck's coming up with a compound for COVID treatment. I worked on that compound. So now I know at some point, we've done that effective work with those. I worked on some of the juvenile animal models to, in which those compounds are now effectively helping the life of millions of kids across the world. So that's kind of how this is called the tunnel effect. You start with a bigger bunch of compounds and drain all the way to a single compound. This would take years. This would take several years, 10 to 15 years, worth investing 60 to maybe couple $100 million. So it's a phenomenal path that it takes to come here. And this is what I told you earlier about the clinical studies. So you start at the preclinical, then you go to phase 1 and you use tens of humans, mostly safety studies. Then you go to these are all healthy volunteers. Then you go to a smaller disease population to do a dose finding efficacy study. Then you move on to a confirmation full study with thousands of disease volunteers and healthy patients and placebo group and then the affected group and so on and so forth. So that's kind of how this thread line actually flows. And these are the timeline that I briefly talked to you about. It takes about 1 to 6 months for a phase 1 study, 3 to 18 months, which is a year and a half, and then up to 3 years for just the phase 3 study alone. So this involves enrollment of volunteers. So this is actually for a disease specifically called the FOP, which actually stands for a specific type of fibrodysplasia type of disease, in which you will see it's actually a rare connective tissue disease. So the FOB stands for fibrodysplasia ossificans progressiva. It's actually a connective type of disease, connective tissue degenerative disease. And then that's resulted in a lot of, benefiting a lot of folks recently and over several years now. And, this is the approximate timeline. So you can see the number of the number of effort put in by a lot of folks, and also the number of days and years you spend to do it, and also the amount of money that's involved in this process. So this is the entire drug discovery. I've tried to concise it and do it a little bit fast. But if any one of you want to know anything in detail about each of these segments, on the processes, individual testing, tox models, GLP, non-GLP, you probably want to, you might want to reach out to me at some point. I'll be more than happy to talk to you guys. But this is the overarching world of drug discovery. When you say a drug, that's where it started, and this is how it's actually ended, helping the life of a patient. So that's the real impetus for me and many of the other toxicologists to actually get out of bed and work with clinicians and patients. And so that's a lot of safety testing. So when I was in graduate school, I've worked with a lot of Parkinson's, people who suffer with Parkinson's disease per se. I've seen them with those kind of, so that's kind of what pulled me into drug discovery and more related to neuroscience as such. So it's a lot more animal models. I have a few animal models of Parkinson's I've worked on, which is exactly like what you'll see in the patients. Those animals function exactly like those human patients themselves. And I can show you those videos and stuff that I have too, if you guys are very interested at later points. So that's pretty much what I have for you guys today, I suppose. Yep. And if you have any questions, let me know, and I'll be more than happy to discuss with you now. So thank you for your time.
So thank you very much, Dr. Adithiya, Charli. You accepted our invitation within a short span of time. And whatever you have delivered is very knowledgeable and technical, more job-oriented kind of a technical session. So we're happy to have your guest lecture in overall. Presently, I welcome participants to raise the questions. You can unmute and you can question if you have any doubts in this lecture. So we are expecting your kind of interaction, participants. So I find more than 120 participants are there for this guest lecture. So students, if you have any doubts, you can ask.
There's somebody called Akshara who's put up her hand, if I'm correct.
Yes, sir. Hello, sir. So,
Yep.
sir, I just have a general question. Do you think with the help of bioinformatics, we'll be able to replace, avoid the testing on animals in the future?
This is a very good question that a lot of people are trying to ask. I think my personal belief is that that's not going to be possible, because you just can't find out the error in a human body with just computer. You can try to find out to some extent by expediting the drug discovery process, but you cannot replace animal work with humans and computer to decipher whatever you want to do. Because if I'm going to tell you that I'm going to give you a drug that was never tested on anything before, I'm sure as patients, none of you guys want to take it either, right? So animals are probably one of the rich resources to actually find out fundamental differences in outputs. But I think nowadays a lot of bigger software companies and some of my friends per se, actually, they are also working with me from Google and Microsoft and all the other places, who actually want to collaborate with pharmaceutical companies to find artificial intelligence technologies to expedite drug discovery. So if you cut down, say, 3-4 years from a 10-year process, you save a lot of money. So that's probably what you can actually do with software. But I don't think practically, and it's not wise and safe enough to actually completely replace animals with bioinformatic tools. But you can definitely use bioinformatic tools to expedite the process, cut down time and money. But I think it would not justify to directly replace animals.
Okay, sir. Thank you.
Anybody else?
So any other questions from the participants? So those who are trying abroad for higher studies, you like to ask anything related to higher studies? Do you have any doubts in that? If no, can we take some video snapshots from all the participants? So kindly switch on your video. We can take one snapshot of with everyone. Can we start the process now? So participants, kindly switch on your video. And at last, I think Madhumati is going to deliver the thank note. Okay, Madhumita, is it so?
Sir, Sanjana S will deliver the order.
Okay. I'm expecting that after this.
Okay.
So presently, all the participants can switch on your video. So I think one is also taking care of snapshots from SPF.
Yes, sir.
So participants come quickly. So I found only few videos. Okay. Have you taken some snapshots?
Yes, sir.
Okay. So then I think we can conclude. Sanjana, you can carry out.
Yes, sir. Good morning to one and all present here. On behalf of Student Biotech Forum, I would like to thank Dr. Adithiya Charli sir for his enlightening talk today. And I would also like to thank the management, secretary, principal, and our HOD ma'am, Dr. V. Sumita, and all our faculty members for supporting us in making this possible. I would also like to thank our alumni coordinator, Mr. N. Satish, for his support. Last but not the least, I would also like to thank all the members of Student Biotech Forum and also to all the students attending this webinar. Thank you.
Thank you, everyone.
Thank you, Dr. Adithiya Charli, and the participants. So have a great day.
Thank you, Aditya. Thank you for taking your time off, attending.
No problem.
Just giving the students a very good lecture.
Pleasure is mine. Thank you, Sumita ma'am. I will probably talk to you guys when I'm coming back to India at some point. But till then, you guys stay safe and take care and wear your mask.
Thank you, Aditya.
Yeah, take care, everyone. Bye, Dr. Kumar. See you guys.
Yes, sir. So I'll be sending you the feedback form to Dr. Adithiya Charli. Thank you. So participants, I request you to fill the feedback form from your side. Have a great day. So thanks for your patience. Bye.